Hi, welcome. So this is part 2 of the October. Let me write down for you guys. October 2024 Biology Unit 2 paper. So yeah, let's just start with it. So the photograph shows a sperm cell beginning to enter an egg cell. A sperm cell swims by rotating its flagellum, right? Rotating. So which of the following statements about sperm cells are correct? So the first one is sperm cells are diploid. The flagellum rotates using energy. The sperm cells have a streamlined shape to increase resistance. So, as you can see, I've written it down. So sperm cells are haploid, and we mentioned this in the previous question here. Over here, as you can see, the generative nucleus, it divides and forms two male haploid nuclei. Okay? So, sperm cells are haploid. The flagellum rotates using energy. This point is correct. Sperm cells have a streamlined shape to increase resistance. That is wrong. As you can say, increase. No, they have a streamlined shape so it can decrease resistance. All right, that's an adaptation that helps them to swim faster. So the answer is going to be D. Moving on, describe how the nucleus of a sperm cell enters an egg cell. So this question is extremely important, you guys. Okay, I'm going to add a star here. It's important that you remember how to answer this question. And it's important that you mention acrosome. It's important that whenever you have a question, write this as an exam tip. Whenever you have a question about an egg cell and sperm cell, you need to mention the zona pellucida. It is not enough for you to mention the egg cell or the egg cell membrane. No, it's not enough. You need to mention the name zona pellucida. Okay? You can pronounce however you want, but it's very important. So, sperm cells contain acrosome, which releases enzymes. These enzymes digest the zona pellucida. Therefore, the sperm cell's membrane fuses with the egg cell or the oocyte membrane, allowing the nucleus to enter the egg cell. It's important that you memorize this answer because it's a frequently asked question. Here we have the oocyte, okay, the egg cell, right? This here is the egg cell membrane, right? What is it called? We're gonna call it the zona pellucida. And inside here we have like the nucleus and everything. But yeah, just need to remember. We need to remember that. And the acrosome. Oh my gosh, I'm sorry. Let me try and draw it for you guys. Okay, this is like a sperm head. We have over here. You're gonna see like there's just some stuff here, and it's basically found within the head of the sperm cell. It's the acrosome. Acrosome contains the enzyme acrosin. In animal cells, the nucleus can divide by mitosis or meiosis, right? So in the cell cycle, the time between two divisions of mitosis is called interphase. Describe what happens to DNA during interphase. This is also another important question. You have to memorize and you have to create your model answers so you can remember and quickly write down the answer so that you don't have to spend too much time thinking about the answer. So, DNA uncoils in the beginning of interphase. DNA is replicated and then checked for errors. DNA undergoes transcription. That's just it. Very simple and easy. So, name the stage of mitosis that follows prophase. We know that's metaphase. So, a person with Patau syndrome had 3 copies of chromosome 13 in each body cell. This occurred due to an error in meiosis and the formation of his father's sperm cells. So the diagram shows what happened to chromosome 13 during the formation of the father's sperm cells. We can see here we have the chromosome 13 still in prophase, and then here we have meiosis 1, and then we have meiosis 2. Okay. We have A, B, C, and D. Let's see. So a zygote is formed at fertilization. Yep. We know that. So explain how an error in meiosis could result in the production of a zygote with 3 copies of chromosome 13. Okay. So in meiosis 2, the centromere does not divide. As we can see right over here, the centromere in this one did not divide. Therefore, the sister chromatids did not separate, resulting in sperm D in containing 2 chromosomes 13S, and sperm C with none of the chromosome 13S. Therefore, the egg cell contains 1 chromosome 13S since it is haploid. Therefore, the male and female nucleus are fused together. So if one cell contains 2 and the other cell contains 1, you add them both, you get 3 chromosome 13S. That's pretty much it. Very simple. Moving on. The zygote would develop into a morula and then into a blastocyst. So which row in the table is correct? We know that the zygote is totipotent and the cells in the blastocyst inner mass are pluripotent. So the answer is C. An individual can also develop Patau syndrome if part of chromosome 13 becomes attached to a different chromosome during prophase 1. Suggest a reason why part of chromosome 13 can become attached to a different chromosome when a gamete is formed. So we know that, this occurs when there's a translocation error. So when a translocation error occurs, this is what happens. So methylation of DNA can alter the expression of certain genes on chromosome 13. Okay, so methylation of DNA can be passed on during mitotic cell division. Explain how DNA methylation can alter the expression of some genes. So methyl groups are added or removed. So methyl groups are added to a gene, okay, or a cytosine. Therefore, the gene becomes switched off or deactivated. So when you're tampering with the methyl groups in a gene, basically what methyl groups do is that they switch the genes off. They deactivate the genes, causing them to become inactive. Okay, so that's it. Explain why epigenetic changes to genes, such as DNA methylation, are passed on by mitosis. So epigenetic changes are replicated. Therefore, daughter cells will contain the same DNA methylation after cell division, since mitosis produces genetically identical cells. Okay, so I think it's pretty easy to understand. If you're having some struggle with this, I would suggest to go over DNA, mitosis, meiosis once again. It'll help you guys out. Now we have question 7. Cellulitis is an infection of the skin. It is caused when bacteria living on the skin surface enter through a cut in the skin. This results in swelling, redness of the infected area. Images show a patient with cellulitis in one leg and one of the types of skin bacteria that cause cellulitis. So this here is the diagram showing the cellulitis, and this is the bacteria. Okay, so this type of bacteria has a capsule, but not a flagellum. So just one advantage of having a capsule to this type of bacteria. So the first one I wrote is that it prevents the bacteria from drying out. Second one is that protection from getting digested by enzymes. So when a bacteria cell has a capsule, it makes it more resistant. It makes it more strong. It basically acts as like a shield of armor, therefore protecting it from getting digested by enzymes, or preventing it from being detected by the immune system, preventing the bacteria from drying out or dying out, So yeah, this is the advantages of a capsule. the second part is suggest why this type of bacteria does not need a flagellum. So this bacteria does not live in a liquid environment. As mentioned here in the question, it was mentioned that this bacteria lives on the skin surface, so it does not need a tail to swim around and move around the skin. Moving on, so which of the following structures would be found in a bacterial cell? So we know that bacterial cells, they do contain a cell membrane, they do contain cell wall and ribosomes, therefore the answer is D. So part B, the antibiotic flucloza, sorry, you guys, the antibiotic flucloxacillin is the current treatment for cellulitis. So a student investigated the antimicrobial properties of the antibiotic. So an agar plate had the type of bacteria which causes cellulitis spread over the surface of the agar. 3 discs containing the same concentration of flucloxacillin were placed onto the plate as shown in the diagram, incubated for 24 hours, 25 degrees Celsius. Okay, here we have the zone of inhibition, disc containing the antibiotic. So the table shows the results. So we have the formula for standard deviation right over here given to us. It's been given to us. So the table shows some of the calculations done by the student. Calculate the standard deviation for the set of data. Use the student's table and formula to help. Okay, so easy. First, we calculated the data that's right over here, which is x minus the mean of x. X here is 7.5. The mean here was given to us 9. Therefore, it's going to be 7.5 minus 9 gives us negative 1.5. And then we just have to square this value. So we get 2.25. And then we find out the sum of the x minus mean of x squared. We just add all of these up and we get 6.5. Okay. And then we just use in the formula. So S is equals to the sum divided by n minus 1. So 6.5 divided by 2, which equals to 1.8027. That's the answer over here. So which of the following explains why the student would want to calculate the standard deviation for this experiment? We know that it's to see how widely the diameter values differ from the mean diameter. The effectiveness of treating cellulitis with the antibiotic alone was compared with a treatment that used both the antibiotic and clindamycin. So the diagram shows how 2 groups of patients with cellulitis were selected from an initial group of 2,444 patients. So we have the initial group right over here. Then we have patients who are excluded. We have a selected group of 420 males with cellulitis on one leg. And then group A has 207. Group B has 203. Okay. So what percentage of the initial group of patients were placed in group B? Basically, what we're going to do is we're going to take the value of 203 divided by the total, multiply by 100, and we get 8.3%. So just how the study could be carried out as a double-blind trial. Double-blind. Use diagram to support your answer. So neither the patient nor the doctors would know if the patients were taking just the antibiotic or both the antibiotic and clindamycin. Clindamycin, you guys, I'm tired. Please don't. You guys understand what I'm saying. Basically, that is what a double-blind trial is. The doctors do not know. The patients don't know either. Over here, they said to not mention placebo or anything like that. So, yeah, if you ever get a question, just to be on the safe side, just mention that neither the patient nor the doctors know which medicine or which pills the patients are taking. So now we have part 3. The table shows the mean difference in circumference for the infected and non-infected legs at 5 and 10 days after treatment began. So we have group A, we have group B. Okay, so mean difference reduces in both over time. We can see that here to here there was a reduction, here to here there was also a reduction, right? That's like an easy in general point we have. And group B had the greatest mean difference compared to group A. You basically subtract these values, and once we do that, we can tell that yes, group B has a greater, it has the greatest mean difference compared to group A. Therefore, treatment for group B is going to be more effective. So these are just like, points that we could tell from the graph. Now, during these questions, what else you can mention are points which were not mentioned within the experiment, or just, information that we could have used to make this investigation or this oh my gosh, experiment more valid, more reliable. So let's see. So the first point I mentioned was that we aren't aware of the initial measurement or if one antibiotic reduced the swelling faster within the first 5 days. We weren't given much information basically about the antibiotics. So, if we had like an initial measurement, that would have, of course, helped us out much more. But we directly just started measuring after 5 days, after 10 days. There was no initial recording. And there was no mention of which antibiotic was used, if it was just one or if it was both of them. None of that was mentioned. And there weren't any standard deviations, therefore we can't judge the significance. That's pretty it's understandable. And only males were used, therefore we cannot apply these conclusions to females. So it is limited. You can just always, whenever you have these kind of questions, comment on the results of this investigation. On the results, you can always mention points. Sometimes it's like we're not sure if the sex was kept as a variable or if their age was kept as a variable, and just different points like that. And I think that's the end for question 7. Thank you so much for watching. Good luck, you guys. Good luck on your exam. Okay, I hope it goes by well. Again, past papers, you don't have to solve, you don't have to practice. What I do, like the last day or like, the last few hours before the exam, I'd open up my laptop, my PC, whatever, and just like divide. So like half of the screen was like the question paper, the other half was the marking scheme. And I just really roughly go over through them both. So yeah, good luck. You guys are gonna do well. Thank you so much for watching. And, send this video to your friends. Like, maybe it'll help them out. And subscribe, because I'm gonna keep posting more videos on Unit 2. And I'm gonna post the rest of this question, the last question here. So just, subscribe and like, click the notification bell so that you guys know when I'm gonna be posting it. Yeah, that's it. Thank you so much for being here, and good luck with your exam. Bye. Okay, cortical granules in the egg cell contain enzymes. These enzymes prevent more than one sperm from entering the egg cell. So describe the role of rough endoplasmic reticulum and the Golgi apparatus in the production of these enzymes. This question, you guys, is also extremely important. So please write down a model answer, memorize a model answer, and use it in your exams. Save yourself some time. Save yourself like the energy. I feel like in bio, if you really want to do well and get full marks, which by the way, I just want to mention, I got full UMS marks in my Unit 2 biology paper when I did it. And I studied everything by myself. So yeah, Yeah, I know. I'm just saying, you guys, write down model answers and just memorize them, because this is another question which is repeated a lot. So let's start with the answer. So we know that translation right here, occurs at the RER, which is the rough endoplasmic reticulum or the ribosome. And then what happens is that 3D structures are then formed within the RER. And then the protein that's formed is packed into vesicles by the RER. And then these vesicles fuse with the Golgi apparatus. And within the Golgi apparatus, a carbohydrate or a phosphate group is added to it. So, okay. We have, this is mostly from what I remember, my memory, guys. This is a ribosome, right? And inside of this ribosome, we have, polypeptide chains, right? We have a chain, and this chain is going to basically go ahead and form like a 3D structure. Sorry, I don't know how to draw a 3D structure, but it's going to create a 3D structure, right? It's going to produce a protein. And ribosome here produces a protein. I'm going to just write as protein. I don't know how to draw it out, you guys. Okay, protein. And then protein is packaged into a cute little, vesicle, right? This is our vesicle. Okay, and then the vesicle moves around in the cell, in the cytoplasm, and it joins the, the Golgi apparatus. You guys know how to draw it, right? It's like a Wi-Fi sign thing. Please ignore my drawing. But yeah, you guys get in, like, you have some ends here. Yeah, that's pretty much just the process, right? You have to memorize it. Ribosome, translation occurs, 3D structures are formed, you get a protein. This protein that's formed is packaged into a vesicle. Vesicle is then transported in the cytoplasm, and it fuses with the Golgi apparatus. Within the Golgi apparatus, a carbohydrate or a phosphate group is added to the protein formed, and that is how these molecules are formed. Okay, move on. So just how these enzymes will prevent more than one sperm nucleus from entering the egg cell. So cortical granules fuse with the cell membrane and they release enzymes, which results in the hardening or thickening of the zona pellucida. Again, you have to repeat the name. It's written in the marking scheme. They do not accept egg cell membrane. They do not accept any other name. They want you to specifically mention this name right over here, therefore preventing acrosome enzymes from digesting the zona pellucida. So we have our egg cell right over here. And then we have cortical granules right over here. These are my cortical granules. My cortical granules are moving to the zona pellucida, okay, to the edge. Over here, a reaction occurs. So now, what happens is that the zona pellucida starts becoming hard, like it starts hardening. Let me choose a different color. We can go with the red crayon, because why not? Oops, okay. It's hardened, and, they cannot pass through. The acrosome cannot digest its way through this thickened wall. Okay, so that's pretty much what goes on in this process over here. Let's move on. So now we have question 5. The Grandidier's baobab is endemic to Madagascar. Fruit bats transfer pollen from one baobab tree to another. Okay, fruit bats from one to another. So the population of fruit bats in Madagascar is decreasing. The photographs show one Grandidier's tree in a field of rice and a Madagascan fruit bat. So here we have the tree and here we have the bat. Okay, so it says part 1 says, this species of tree is endangered. So identify 2 reasons why this species of tree is endangered. Use information above and the photograph to support your answer. So I started with there's a lower pollination rate due to a decrease in the number of fruit bats, right? If I have less number of fruit bats, I'm going to have like a less number of animal, which is carrying the pollen from tree to tree. Therefore, there's gonna be less reproduction occurring. So that's, the first point that I think is really easy to understand. Next point I wrote is that more number of baobab trees are being cut down to grow rice or crops. So as more number of trees are being cut down to grow these rice and crops, the population of the tree is decreasing and lowering. Third point is that the trees are far apart from each other, therefore there'll be less chances of pollination, right? If I have a tree that's over here and another one here, it's very easy to move from one tree to another, as you can see. But if I have a tree that's right over here and I have another tree that's all the way over here, it's not that easy for animals. Animals maybe might go here, they might go all the way here or here, but fewer animals are gonna go all the way here to this other tree. Therefore, we're gonna have less reproduction occurring within the trees. So now let's move on to part 2, sorry. So give 2 reasons how education could help conserve the Grandidier's baobab. So the first point I wrote was Madagascan people will be taught to limit or reduce the cutting down of trees. It's pretty self-understandable. Second point is Madagascan people will be taught to protect bats because, the more number of bats we have, the more number of pollen carriers we have. Therefore, more number of trees will be produced. And the last point I've written is Madagascan people will be taught to plant more baobab trees to increase the population. So, yeah, it's pretty self-explanatory. Very nice, easy question. Now, part B. Calcium ions are used to make molecules in plants. So explain the role of one named molecule that uses calcium ions. So the name of molecule is calcium pectate, and the role of this molecule is to increase cell wall stability. Moving on, water is also important to plants. Which of the following would transport water down the stem of a plant? We know that is the phloem. So the photograph shows pods on a soybean plant. The effect of magnesium ions, okay, so now we're talking magnesium, on the number of pods per soybean plant was investigated. In 2019, soybean plants were grown in 2 fields. So one field had no magnesium ions added to the soil. We have no magnesium. And this other one had magnesium ions added to the soil. The mean number of pods per soybean plant was determined for both fields, okay? And then we repeated the same experiment 2020 and recorded the results. So let's see. We have the graph over here. So give 2 conclusions that can be made using the data. First conclusion that we can make is that there were more pods per soybean plant in which year? 2020 than in 2019, right? As you can see. And then second point is that there were more pods per soybean plant when magnesium ions were present. Here we have no magnesium. Here we have magnesium, no magnesium, magnesium. So we can see that, yes, there is a difference and an increase. It's pretty self-explanatory. So next part, in another investigation, the effect of magnesium ions on the mass of soybeans and maize grains was studied. So now we have soybean and maize. So graph shows the results, as we can see. Let's move on. It says comment on the results from this investigation. So the first comment is the presence of magnesium ions increased the mass for both. As we can see over here, there was an increase. Here, there was an increase. So yeah, the presence of magnesium ions definitely did cause an increase in both soybean and maize. So the next point I have mentioned is the mass of maize is greater than the mass of soybeans. As we can see over here, a greater number of maize was grown. than compared to soybean. Grown, produced, whatever you want to call. And then the third is that the percentage increase is greater in soybeans, as you can see here. You can see we have a difference of about 2 blocks. Here we have a difference of, it's almost the same. Actually, no, it's just 1.5 block. So yeah, it was much greater than in soybeans than it was in maize. And the difference is significant between maize and soybeans due to no overlapping of the error bars. As you can see over here, the error bars here and here are not overlapping. Therefore, the results are significant. And I think that's the end. Next question.